Hi, welcome to class today. In our lesson today, we're going to discuss scientific inquiry. What we mean by that is posing scientific questions, setting up controlled experiments, and then based on the results, drawing conclusions. For our lesson today, we're most concerned with making observations and then posing questions about those observations. So our objectives for today's lesson include describing the steps involved with scientific inquiry, and differentiating between an observation and an inference. Now, these last two objectives we're going to get to in a later section for this lesson, and they involve how to set up controlled experiments and the difference between scientific theories and laws. So, scientific inquiry. As you can see here, there's several steps involved with setting up a scientific question, setting up an experiment about that question, and then eventually drawing a conclusion. So as I mentioned, in our section today that we're going to cover, we're most concerned with how to make scientific observations about the world around you, and then how to properly set up or pose a scientific question about those observations. So the remainder of our steps to scientific inquiry, we're going to talk about in a later section for this lesson, and those are coming up with a testable statement, which is called a hypothesis, setting up a controlled experiment, analyzing the results from that experiment, and then finally, eventually, you're going to formulate a conclusion about that experiment. So for today, we're just getting started with, first of all, how do you make observations properly, and then how do you come up with a question that you could pose about those observations that you would later conduct an experiment about. So, making observations. What that means is using as many of your senses as possible about the natural world. So, what that would mean as far as using senses. What do you see? What do you hear? Can you touch whatever it is you're observing? Can you smell whatever it is you're observing? Can you taste whatever it is you're observing? Okay, of course, that's going to depend on the type of experiment and what it is you're working with. But you want to, again, use as many of your senses as possible and use as much detail as possible when you're making those observations. For example, you can see here this child is looking at something to make an observation. He wants to write down as many observations as he can about what he's looking at in as much detail as possible, so that if somebody else were to look at his notes about this observation, they would have a picture in their mind of exactly what he was looking at. That's how detailed you would want to be. There's two categories to observations. There's qualitative and quantitative. Qualitative would be a description that does not deal with any numerical value. For example, let's say you were looking at a garden and you were observing flowers within that garden. So the color of the petals, for example, would be a qualitative observation. A quantitative observation, on the other hand, is numerical in value. So with that same example with the flowers, let's say you count the number of petals. So now we're dealing with numbers, so that's a quantitative observation. All right, now, based on observations, you can also infer about those observations or make an inference. What that means is you're using prior knowledge about the subject to create a possible explanation. So let's say that you look outside and you observe that it's cloudy and that there's storm clouds rolling in. So your explanation would be, okay, it's about to storm, I better bring an umbrella with me today. So that would be you inferring about your observations, making an inference. You're using your prior knowledge, you know from the past that if you see storm clouds rolling in, there's going to be a storm, there's going to be rain, you should bring your umbrella. So you're making an inference about what you observed. All right, now, based on your observations, you're going to want to come up with a scientific question that you could eventually run an experiment about. So that's what we mean when we say testable. Can it be answered with experimentation and observation? So So we need to be as specific as possible when we're posing a scientific question so that in our experiment, we're able to test just one factor or one variable at a time. If we have too broad of a topic, there'd be too many different variables to consider, and you wouldn't know why you got the results you did. So you always want to try to be as specific as possible. So for example, let's say you're observing some birds and they're building a nest. Now, when you come up with so that's your observation, but when you're coming up with your question about that observation, you may need to do some research so you can narrow down your question. So you could research things like the type of bird that you observed making the nest, the plants that they were using, or the tree parts that they were using to make their nest. So the different parts that are involved with their nest. By doing that type of research, now when you pose your scientific question about this observation, you're narrowing down, why does a robin use this specific type of plant when building a nest? Okay, rather than just saying, why is there a bird building a nest using parts of a tree? So again, the more specific you are using research and using observations, the easier it's going to be to actually run an experiment about your scientific problem. All right, so thinking ahead, as you can see here, we've got some yummy looking chocolate chip cookies. So let's say we were to set up an experiment to bake ourselves the ideal chocolate chip cookies. So thinking ahead would be, how could you set up a controlled experiment to get the perfect chocolate chip cookies, baking them for the ideal amount of time? So let's think about, how would you set up your experiment and be able to collect data about that experiment so somebody else could look at it and go, oh, okay, I get it. This is what I should do to bake perfect chocolate chip cookies. That's thinking ahead to the next section of our scientific inquiry lesson. Hi, welcome to class today. In today's lesson, we're going to continue our discussion of scientific inquiry. We're going to talk about, how do you properly write a scientific hypothesis based on your observations, and then how do you set up a controlled experiment based on your hypothesis. So let's go ahead and take a look at our objectives. We're continuing with our discussion of the steps involved with scientific inquiry. So remember, we previously discussed, how do you form scientific observations and the difference between that and an inference. And we talked about, how do you pose questions about your observations. So now we're going to talk about, based on those observations, how do you formulate a hypothesis? And then also, how do you set up a controlled experiment? Now in that experiment, we're going to have variables. We're going to have the independent and the dependent variable. And for it to be controlled, that means we need to keep all of our factors constant other than what it is we are testing. So we need to have what's called the control group and the experimental group. All of that will be discussed because that is what is part of a controlled experiment, meaning we know that our results are going to be valid in the end. Our last objective we can't get to until after this discussion, because that involves theories and laws, which we would never have without these controlled experiments and conclusions of these experiments. So our last objective we'll get to in the last section for scientific inquiry. Okay, so let's kind of take a look here at our map of the steps of scientific inquiry and where we left off. So we previously discussed how to form scientific observations using your senses in as much detail as possible, how to pose scientific questions based on those observations. Now today we are ready to discuss, how do you then formulate a scientific hypothesis? One of the things we're going to talk about is, your hypothesis is going to be an if-then statement, and it must be testable because we have to be able to set up an experiment based on our hypothesis. So we'll take a look at an example of that today. And then we're also going to go through the motions of setting up a controlled experiment. So we'll talk about the variables in an experiment, what the controls are in the experiment, so that we know that we're getting valid results based on just what we are testing. And then our last two pieces of scientific inquiry will be discussed in our last section, because eventually we need to know how to analyze our results so we can form a conclusion. So let's start with our first objective for today, which is how do we formulate a hypothesis? All right, well, a hypothesis is an explanation for whatever it is we've observed. A lot of times you're going to have prior knowledge on this subject, and you will have researched the subject because oftentimes it's something that you're already interested in, or maybe you've done previous experiments with a related subject. Okay, so usually you're somewhat knowledgeable about this subject before you actually formulate your hypothesis. It's different from a prediction. We're not simply stating what we think will happen. We need to be able to test what we're saying in the hypothesis. We need to be able to set up an experiment. So it's a testable prediction. So it's tested with experimentation, and you could have additional observations involved with your hypothesis. It's going to be an if-then statement. So we're going to use a pretty simple example that probably everyone's familiar with and can easily follow. Our example is, let's say we want to bake chocolate chip cookies, and we're thinking about the relationship between, okay, if we're adding more and more chocolate chips, what's that going to do to the baking time so that we get our perfect chocolate chip cookie in the end, right? Because, of course, we always want to get that perfect cookie to eat when we're all done baking them. So we're going to formulate a hypothesis around this relationship between the number of chocolate chips and the baking time. And again, we need to formulate it in a way so that it is testable, so that we can set up a controlled experiment about it as an if-then statement. So for example, we could say, if we increase the number of chocolate chips, our hypothesis is we will then have to increase the baking time. So what we're suggesting is, by increasing the number of chocolate chips to get your perfect cookie, you have to keep increasing the baking time. So we've written it as an if-then statement, and it's definitely testable, right? We could get into the kitchen and we could test this for ourselves. We could set up a data table where we list how many chocolate chips we're going to be adding for different trials, and then we could also write down what is happening to the baking time as we're doing that. Okay, so we followed our steps. We've set up a testable statement, if then. Now let's talk about how could we set up our experiment about this statement. So creating an experiment. Well, we need to have variables. So in this case, we have two. We have the number of chocolate chips and the baking time. Now what you need to ask yourself is, what depends on what? So you have the independent and the dependent. The dependent variable always depends on the independent. So our independent in our example is the number of chocolate chips, and our dependent is time. Well, how do I know that? Let's think about this. Our baking time for our chocolate chip cookies depends on the number of chocolate chips we're adding, right? Because what we suggested in our hypothesis is, as we increase the number of chocolate chips, that's causing our baking time to increase. Okay, that's what we just suggested in the hypothesis that we wrote. So your dependent variable always depends on the independent. Our number of chocolate chips is independent, right? We're just adding those, but our baking time depends on how many we're adding. Okay, so that's for our experiment that we're using as our example. Those are our variables. Now, we also need to have controls in our experiment. What do we mean by controls? Well, we need to keep other factors constant, so we're sure that it's the number of chocolate chips that's causing the change to our baking time. So for example, we could say one control in this experiment would be to use the same oven for all of your trials, right? We don't want to change and use different ovens because we don't know if that's affecting our baking time. We would also want to keep constant all our other ingredients. Okay, because again, we're only dealing with our chocolate chips as our ingredient and the effect on the baking time. We can't be messing around with the number of eggs or the baking soda or the flour or whatever. Everything else needs to be kept constant so that you know in the end the number of chocolate chips was what affected the baking time. Okay, that's the idea with controls. So I mentioned also a control group and an experimental group. So we could say in this case our control group would be to run a trial without any chocolate chips at all, and then an experimental group could be, or there'd be several experimental groups as we're adding more and more chocolate chips. So you always have a control group to use as comparison, and then your experimental group as you're experimenting with your variables. All right, also in an experiment, it must be repeatable. Okay, so in this case, yes, we could run this experiment several times and see if we're still getting the same results. And it must follow a procedure. So you must be able to write down exactly what you should do so somebody else could then refer to it and follow that same procedure. Includes a control group and an experimental group. So again, as I mentioned, your control group is used for comparison so that your experimental group, you can compare the results you're getting to your control group, which is just under normal conditions. All right, so thinking ahead, so we have this beautiful plate of chocolate chip cookies. So let's say we went ahead and ran this experiment. Well, thinking ahead, how would you record your data for this experiment? And then how could you display that data so it could be analyzed? And then how could you form a conclusion about this experiment? So all of these things will be discussed in our last portion of scientific inquiry so that we can finally determine, well, how is it that we come up with scientific theories and scientific laws? Okay, so in our last section of scientific inquiry, all of that will be discussed. Hi, welcome to class today. In our lesson today, we're going to continue our discussion of scientific inquiry. We're going to talk about, once you've conducted a controlled experiment, how do you record and analyze your data from that experiment, and then eventually, how do you formulate a conclusion? So for today's objectives, we're going to continue describing the steps involved with scientific inquiry. We've previously discussed the difference between an observation and an inference. We've also previously discussed how to set up a controlled experiment with a control group and an experimental group, and also with independent and dependent variables. The last piece we're going to discuss in today's section is, after you've run that experiment, analyzed your results, formed your conclusion, well, how do you ever end up getting what are called scientific theories or scientific laws? How do those come to be from different experiments, from conclusions of those experiments? So that's the last piece we need to talk about for scientific inquiry. All right, let's kind of review here where we left off with our steps and what we're going to discuss today. So remember, we've previously discussed what it means to make scientific observations, remember, using as many senses as possible in as much detail as possible. We've also talked about how do you pose a scientific question based on those observations. How do you formulate a hypothesis as an if-then statement and a testable statement? How do you set up and run a controlled experiment? That's where we left off. Now, what we're going to discuss today is, once you run that controlled experiment, how do you record your results and analyze those results? Once you do that, how do you formulate your conclusion? Then the last piece that kind of goes along with conclusion is communication. You would communicate those findings to the scientific community, because remember, again, somehow we end up with scientific theories and scientific laws. Well, if there was no communication within the scientific community, I don't think those laws or those theories would ever exist, because nobody would know about all these experiments and nobody would take a look at the findings, build upon those findings, run more experiments, and the knowledge keeps growing from there. So communication is a big piece after formulating a conclusion. All right, so let's start by discussing how to analyze data at the end of an experiment. So remember when we talked about observations, and we said that observations could be qualitative or quantitative. Well, the same goes for data. Remember, qualitative meant if you're describing something, but it is not in number format. It is not numerical. So for example, the color of a substance, if you were to record that kind of data, you record the color of the substance, that would be qualitative data. Quantitative, on the other hand, is numerical values. So if you're recording the mass of a substance, you're recording a number. So that would be quantitative data. Now, as you're making or recording these observations or recording these pieces of data, you want to do that in an organized format. So you're going to organize it according to the type of data. So you could organize it according to quantitative or qualitative. You could also organize your data in terms of the variables, independent, dependent variable. The whole idea here is organizing it so that if somebody else were to take a look, it would make sense to them why it's organized the way it is, and it would help them to analyze the data. So a few different ways you could do that. The first way would be to take whatever it is you've recorded during the experiment and put it in an organized data table format. We'll take a look at what a data table might look like here in a moment. Also, you could then take your data table and you could make a graph based on that data. So that could be a line graph, a bar graph, or a pie graph. So we're going to take a look at examples of each of those and how it helps to represent your data in those formats. So that again, if somebody took a look at those graphs, it would make sense to them what those graphs are representing. So let's talk about data tables for a moment. Let's say we're going to have a data table to compare the independent and the dependent variables. Okay, let's talk about using the same example from the previous section. Remember, we were talking about baking chocolate chip cookies, and we were saying that the number of chocolate chips and the baking time were the two variables. So the baking time depended on the number of chocolate chips. So our independent variable was the number of chips, and our dependent variable was the time, right? Because the time was changing as the number of chips were changing. So here's what your data table could look like for that same experiment. We have the number of chocolate chips as our independent, cooking time is our dependent. And you can clearly see in this table format the relationship between the two. As the number of chips are increasing, the cooking time is also increasing. If we didn't put it in a table format, let's just say we wrote it out in paragraph format, I don't think this relationship would be as clear to somebody that was just reading through the information, right? It's a lot easier for them to go, oh, okay, I see the relationship. These numbers are increasing, and so are these. If you wrote it all out as a paragraph, I don't think it would be as clear. You might still come to that conclusion, but it wouldn't stand out to you right away. So that's the whole point of having it in this organized format. All right, let's say then you want to take that data and you want to put it in a line graph. Well, a line graph is good to compare the quantitative independent and the quantitative dependent variables. So this experiment is a perfect example of when to use a line graph. Something to remember about graphs: your independent variable is always going to go on the x-axis, and your dependent variable is always going to go on the y-axis. So let's take a look at what our line graph might look like for this experiment. First of all, notice there is a title at the top. That is very important. If somebody saw this same graph in a lab report and no title was attached to it, you would have no idea what it was you were taking a look at. It would just look like a line with a bunch of numbers. So you always want a title at the top so it stands out right away. Okay, this is what this graph is representing. Now we have, remember, our independent variable is the number of chips. You always want to label that, and again, that goes on the x-axis. And then our dependent is time, and you want to be as specific as you can, so you should indicate the time is in minutes, and that goes on the y-axis. Now, this graph is a lot easier for somebody to take a look at, understand, analyze, and interpret. Okay, what about a bar graph? Using our same experiment. Well, a bar graph could be used to compare quantitative or qualitative independent variables. In this same example, of course, we're dealing with a quantitative independent variable, the number of chocolate chips. But oftentimes in bar graphs, it's a qualitative independent variable. For example, let's say you were making a bar graph of different eye colors or different hair colors. So each bar represented a different color, like hazel, blue, or brown. Okay, that would be an example where on your x-axis you have qualitative data. Now on our y-axis for our dependent variable, of course, that's still going to be quantitative, because let's say you were doing the number of people that had those hair colors. So again, on your y-axis, the numbers would be quantitative. back to our example with the cookies. Here's what our bar graph could look like for that example. Again, we have a title at the top. We have our number of chips on our x-axis as our independent variable. It's already labeled here. Each bar is labeled with the number of chips. And then remember, we have our time, and our y-axis looks like this time is actually in minutes and seconds, so it's a little more specific. But again, if you were to put this in your lab report, everything is labeled and clear for somebody to analyze and interpret. Okay, the last type of graph is a pie graph, and the example we're going to look at is not related to our experiment because it's kind of difficult to do a pie graph for our cookie example. So we're going to take a look at a different example just so we understand what a pie graph is. Basically, it's used to compare parts to a whole. So it's basically just a circle. There's sections within that circle to compare parts to the whole entire circle. So in this example, the title is Global Population. So this is a good way to represent by region a comparison of the different populations. So you see there's a bunch of different regions already labeled and color-coded on here, and the different increments on our pie graph or on our pie chart are a good way to visualize if the whole chart represents the world, how many regions are there within that world, and within those regions, what is the population? Okay, once we analyze our data, then we're ready to formulate a conclusion. So when you're formulating a conclusion, you're always going back to your hypothesis. Was it or was it not proven? That's the whole point of doing the experiment. Remember, we wanted a hypothesis, which was a testable statement, so that when it comes time to draw conclusions, we can go back to that statement and say, okay, did I prove it to be correct? Yes or no? How can you prove that? Well, that's why you recorded your data and analyzed your data, because you always need concrete scientific proof to support what you're saying in your conclusion. Your conclusion can be communicated in written or spoken format. Again, very important to communicate your findings to the rest of the scientific community. This is how, even if you did not prove your hypothesis to be correct, somebody else could take a look at those results and it could make them consider a new hypothesis. And that's how scientists build upon each other's findings. That's how eventually we end up with theories and laws. The whole scientific community builds upon each other. With our chocolate chip cookie example, probably if you were working in a bakery, you could communicate your findings to your coworkers so that everybody's on board as to how to properly bake those cookies so that you're getting more people to buy them, for example. Okay, so we could be talking small scale or large scale here, but it's always important to communicate your conclusion. So again, with our chocolate chip cookie example, you'd be communicating, okay, what was the relationship between the number of chocolate chips and the baking time? That's what you would be discussing in your conclusion. All right, finally, our last piece here for scientific inquiry, theories and laws. Well, okay, we're getting these. Remember, at the end of our experiment, we have our conclusion. Now, if we confirmed our hypothesis, so we proved it to be correct, eventually, after many experiments, usually there's revisions along the way, but after many experiments and many confirmations, you can get what's called a scientific theory. So that's an explanation for a set of events or observations. An example would be the theory of evolution. It can be modified when necessary. The theory of evolution is not something that's just concrete. It is the way it is, where there's never going to be new evidence to support or reject it. It just is what it is without a doubt, right? Because the theory of evolution is something that scientists continue to debate and discuss and find evidence for and all that sort of thing, right? So a scientific theory can be modified. Scientific law describes what occurs every time in a specific situation. The law of gravity, for example. What goes up must come down. I don't think that's going to change here on planet Earth, okay? So the law of gravity is not going to be modified. It is what it is. It is a scientific law. Everybody knows that as truth within the scientific community. So that's the difference between a scientific theory and a scientific law. And again, remember, These laws and theories would never exist without the different experiments that scientists conduct and without their hypotheses that they continue to revise and change and experiment with. We would never get to this point where you have theories or laws. Okay, mission accomplished. We have discussed all of the pieces now for scientific inquiry. Real quick, let's review. We talked about it all starts with observations, using your senses, writing down detailed information about what you're observing. Then you pose questions about those observations. You come up with a hypothesis because you start thinking about how you might want to test different things that you've observed. So a hypothesis is an if-then testable statement. You set up a controlled experiment with a control group and an experimental group with an independent and dependent variable. You run that experiment. You record your data. You analyze your data. That could be in a table format, or you could have line graph, bar graph, pie graph. All right, all of these are different methods to analyze and interpret data. Finally, you formulate a conclusion. In your conclusion, you revisit your hypothesis. Did you prove it to be correct? Yes or no? Did you use evidence from your experiment? So we're backtracking to our other steps here. You analyzed whatever it was you found out from your experiment. That should go into your conclusion. All right, even if you did not prove it to be correct, you still want to explain why in either case. So you always want to go back to what happened during the experiment. That's the whole reason you write down your data and you analyze your data. So now we've covered all of the steps involved with scientific inquiry, and you are ready to start running your own controlled experiments as scientists.